Good evening, everybody, and welcome to IF Oxford, the Science and Ideas Festival. It's lovely to have so many of you joining us this evening. Tonight's event, Ocean Reactions. Why should we care about ocean chemistry? Well, to help us understand why we need to care about ocean chemistry, we have two speakers with us tonight. Russell Arnott is from Incredible Oceans, and Kolisa Sinyanya is from the University of Cape Town and is also representing the Ocean Women Fellowship. So without further ado, I will pass over to Russell and he can start our session this evening. Russell.
Cool. Thanks very much, Kathy. So it's going to be me and Kolisa. We're going to have just a bit of I guess like an informal chat about some of our research and how that relates to ocean chemistry. And then obviously we'll have some Q&A from you guys. So I'm going to start off with a little introduction to my research. So I am a marine biologist also at the University of Bath, and I work with Lee. And I guess my background's in marine physics, and I've been very interested in how physics impacts on other systems within the oceans and lakes and reservoirs and things. I always got a bit frustrated, I guess, when I was studying at university, that it was like, if you are a physicist, you must only study the physics, which is like waves, tides, currents and things. And obviously it's it's like, well, if you're just studying one thing in isolation, there's so many impacts and things and knock-on effects and feedbacks. It's actually really interesting to take one area that you're trained in and try and apply it to other things. So my research at University of Bath is looking at how physics, specifically turbulence and mixing, impacts on phytoplankton. And phytoplankton are tiny single-cell plants that live in all of the world's waterways. And like all plants, they require nutrients to grow. So in terms of the chemistry that I'm interested in, I'm very much focusing on those nutrients and the nutrient availability that is going into those phytoplankton cells. So what I do, and I'm going to share my screen now. So I use these things. These are called mesocosms. And that's a posh science name for basically a giant tank. You might have heard of a microcosm or a macrocosm. These are in the middle. They're about 5 meters tall. So this is the bottom level and the top level. And each one holds 2 tons of water. And these are based in the University of Umeå, which is over in the northernmost city of Sweden. And what's great about these is you can tweak what's going on inside them. So if you were to go offshore in a boat, then you measure what's happening, and you get what you're given, kind of thing. Whereas here, I can tweak all of the different nutrient amounts. I can tweak the physical, like how mixed it is. I can tweak how hot or cold it is at different levels, and I can see how the phytoplankton react to that. So it's really, really awesome piece of kit. So when I'm taking samples, and the kind of like quick and easy ways, you use these nutrient tests, which are like little dip tests, and you just dip those in and it changes color. Problem that I quickly found out is I'm colorblind. So trying to kind of pair up what color and things were, I was like, this is not going to stand up to any kind of scientific scrutiny, this guy with dodgy eyes. So in the end, we did it all electronically, which was quite cool. These awesome sensors and that measure your nitrates, your nitrites, your phosphates, silicate, and all these other different nutrient species that phytoplankton need. So the reason I'm kind of researching this, the reason I think it's interesting, is phytoplankton are brilliant. And I must admit, when I first started researching them, I was a bit of an arrogant physicist, and I thought these are just green floaty things that are floating around. And I've since they've stolen my heart, phytoplankton. This one in particular, this is my favorite one, because when I discovered that this existed, I was like, what is this? So this is what phytoplankton people call a motile mixotrophic ciliate. So motile means it can move and it can swim. Mixotrophic means that it's halfway between a plant and an animal, and I'll explain what that means. And a ciliate, which is a group of phytoplankton species, which are distantly related to jellyfish. So this thing from the top looks like a sunflower, but what it actually is kind of two balls on top of each other with like a ballerina tutu going around the middle of it. And what this thing does when it's just hanging out in the water, it's got those 6 spikes coming out of it, which are like long tentacles, which it puts out in the water to increase its drag to stop it from sinking. But if it detects something moving towards it that is giving off a large enough pressure wave that will cause it to be like, oh God, I think someone's going to eat me. What it does is it wraps those tentacles around the top ball, and then it flaps its ballerina tutu, and it can swim ridiculously fast. So considering that this is the same thickness as a human hair, being able to swim 5 millimeters, jump like 0.9 millimeters at a distance wise, but its mean jumping velocity is 1.2 centimeters a second, which is like, you imagine like you're the same thickness as a human hair. That's like I think that's like 200 body lengths, or so, over 200 body lengths that it can jump, which is, pretty, it's like us being able to high five low flying airplanes. Going on to this like mixotrophic thing, it's halfway between a plant and an animal. That thing there, that little tassel coming up the bottom, is its mouth. And so what this does is it finds other plankton cells and eats them. And as a physicist, I was like, I was not expecting the phytoplankton, which I had been told were all plants, were going to eat each other. And this one's particularly cool because when the cell is first produced, it doesn't have any chloroplasts to photosynthesize. So if you think back to like, your time at school, I used to be a science teacher and was forever getting kids to draw leaf cells, and these have got chloroplasts in. This is something that doesn't have any chloroplasts, but it's still categorized as a phytoplankton. What it does by eating those other cells, it then absorbs the chloroplasts from that and then starts using them. So it steals chloroplasts, which I was just like, this is unbelievable. This blew my mind. So this species in particular is quite interesting because this is what we call harmful algal bloom species. And Mesodynium in particular, they really, really like phosphates. And phosphates are one of the kind of key fertilizers that a lot of farmers put on their fields to promote growth. But then obviously what happens then is it rains, and certain amount of that water then ends up going into waterways, and any excess nutrients that haven't been absorbed by the plant, by the field, the crops that the farmer's trying to grow, gets washed into waterways, and it promotes these massive harmful algal blooms. So this is an actual algal bloom that's happened in the Baltic. So that's the area which I'm studying. And as well as just looking kind of gross, and this is a big problem. It's a big problem for tourism because all of these, no one wants to go swimming in water that's like thick, like pea soup. But on a chemical level, what happens is as this bloom happens, it turns the water so murky that sunlight can't get down into the lower depths. So it reduces any other thing from being able to grow properly. And if you're a fish, it can clog up your gills. Then what happens is once the bloom has happened and the cells have kind of done their thing and used up the nutrients, then the bloom sinks to depth and starts to biodegrade and break down through bacterial decomposition. And that bacterial decomposition then causes these vast kind of oxygen dead zones where it's just water that has got no oxygen in it, which obviously then has a knock-on impact for anything that then needs oxygen, like fish, for example, who then die, and it causes these mass fish kills. So Mesodynium has been seen to form these, and they've seen it's an indicator that maybe other harmful algal bloom species might happen. Another thing that another type of harmful algal bloom are these ones, which are called red tides. Now, red tides happen, and we're still discovering a lot of phytoplankton species, but these about 200 of them out of the, I don't know, maybe 200,000 that we know about are actually toxic. So if you were to go swimming in a red tide, it would probably irritate your skin, and if you've got some in your eyes, it would irritate that. If you swallowed some, you'd probably get kind of nausea, diarrhea, and vomiting. The problem is that these, phytoplankton are the basis of the food chain. And so if you've got anything that is a filter feeder, then it can absorb these plankton toxins. And so this is a case that happened maybe like 3-4 years ago in Alaska, that a harmful algal bloom happened. It's one of these red tides, and the toxin was then absorbed by filter oysters and barnacles. And at that point, the toxin bioaccumulated to such high concentrations that then it killed all the mussels off. And so the mussels are dead because they've absorbed this toxin. So then in come the lobsters, and lobsters are and crabs and things, they're like scavengers, they like eating dead things. So the lobsters came in, ate the dead mussels, and then the lobsters died. And this is a photo of like all the lobsters dead floating on the surface of the water from having eaten that. So then, you imagine each level, it goes up, each trophic level, as it gets higher and higher up the food chain, that toxin is bioaccumulating into higher and higher concentrations each time. So then all the lobsters were dead and floating. In came the sea otters. Sea otters then ate all of the dead lobsters, and then they died because the toxin at that point was such a high concentration. And then at the top of the food chain, we've got orcas. The orcas came in and they ended up eating some of these dead sea otters. And at that point, the toxin was in such high concentrations, it was able to kill a killer whale effectively. And so this is, when something like this happens, ecotoxicologists come along and they take samples. And so what they've done here is they've cut the dead orcas, not pectoral fin, the dorsal fin. I should have remembered that. There we go. Cut that off, and it has. And they're going to take that back to the lab, run some things on it to just kind of see what plankton toxin caused that, what kind of how high the concentrations were. So yeah. So unfortunately, the bad news is that because of global warming, and this is kind of where my physics research comes in, global warming is causing these harmful algal blooms to happen more frequently. And when they do happen, they tend to happen with a higher severity. These now happen with quite high frequency in the Great Lakes and things like that. And my own research here at the University of Bath is looking at how we can reduce these algal blooms from happening in reservoirs. So even like on a basic level, water companies spend billions every year trying to filter out the chemicals that plankton give off from their water. There's a chemical in particular called josmin, which is kind of gives water this musty flavor, and water companies can get fined for it. Incidentally, josmin is the chemical that camels smell, and that helps them find oases in the desert. But humans are particularly sensitive to this, and even our own noses, which we're not used to thinking are that kind of sensitive. We can detect one microgram of josmin in a liter of water. That is how sensitive we are to josmin. So trying to find ways of reducing algal blooms in reservoirs has also a massive impact as well. So that's kind of like my research and my different areas and things that I'm interested in. So yeah, I guess we'll do questions at the end. But if you have got any questions, do let me know. And it'd be really cool, I think, to bring Kolisa in now. And if Kolisa, if you want to kind of be like, here's my research, that'd be pretty cool.
Hi, thanks, Russell. Hi, everyone. Welcome and thank you for inviting me to this Ideas Festival. This is a great honor. My name is Kolisa Sinyanya, and I'm a PhD candidate at the University of Cape Town in South Africa. And today I'm delighted to be talking to you about a session from my PhD. My PhD overall talks about or tries to understand geochemical cycling, and the focus is on nitrogen, but overall nutrients and the role they play in relation to microorganisms. And the microorganisms that I specifically look at are phytoplankton. So firstly, I'd like to tell you who I am, where I come from, because I mean, this is a foreign entity for me. You know, I'm African. You guys in the UK, I know that we have people from South Africa watching. Hi, guys. People from all over the world. But I think me trying to paint a picture of who I am as a scientist, where I come from, my background, will make my case stronger. So my background is that I was born in a very small town in South Africa, the Eastern Cape. The town's name is Mthatha. And actually, I have been given a name, Mthatha Boffin, after this article that you see on your screens came out. I don't remember when it came out, probably 2 years ago. And the writer said, Mthatha Boffin puts ocean under scrutiny. That's my favorite headline of all time, by the way. And soon after it came out, everyone started calling me Mthatha Boffin. And then on Twitter, they started a hashtag. So I have my own hashtag Mthatha Boffin. So remember, Mthatha is my hometown. That's where I come from. It's a university town. There's a university there known as Walter Sisulu University. So that's where I did my undergrad and my honors degree. I don't know if in the UK you do the undergrad and honors. I know in America, it's a bit different, but we do the BSc and then you do your honors and you specialize in whatever you'd like to specialize in. So I specialized in botany, and I was mostly fascinated by the biotechnology side of botany, which dealt with microbes. So I went ahead and studied that, and that propelled me towards studying microbes in my master's. So in my master's, I moved from the University of Walter's University to the University of Cape Town, which is under the very famous Table Mountain, a wonder of the world. And that's why I did my master's, studying microbes that form a symbiosis or a relationship with the fynbos biome. And we know, some people know that the fynbos biome is endemic to South Africa, to the western and northern regions of South Africa. So that's fascinated me. And I was like, well, I think I really like these microbes, because I was also doing genomics, analyzing their DNA. And when you get to see what they do, who they are, the species, when you go to genus level and then you can go to species level, that's really fascinating. So I was like, maybe I want to do this in the ocean. So I went ahead and tried to apply to get into the ocean science. Now, when I arrived in ocean science, I realized that, whoa, okay, first of all, there are very few women in this field, and even fewer Black women. When I enrolled, I was the first Black woman to enroll in the discipline that I was working in. Now there are more people that have come in, and this was in 2016. So when I started, I had this culture shock. Fortunately, when I arrived at UCT, because in South Africa, we have these disparities of these racial disparities of Black and white. And so UCT was mostly a white university in a predominantly Black country. So for me, when I arrived there, that was a culture shock. And then when I got into ocean science, it was even a bigger shocker for me, because I had to adjust, firstly, for being a woman, and secondly, being a Black woman. But I saw that this works for me, actually, because immediately when I started my PhD with the Department of Oceanography at the University of Cape Town, I could say I became, there were articles written about me. I was invited to speak. I decided to to join competitions and I'd win. Not because I'm a Black woman, but because I was good at what I was doing. So with the exposure came even bigger audiences. So I was part of shows, documentaries. There's still documentaries that I'm part of now. I went on CNN at one point where they wanted to feature me as one of the up and coming women in South Africa in the field of science. But one of the most fascinating things that I have done is to be created as a super scientist. So there's a character in Xhosa we say, Xhosa is my home language. Xhosa we say upopai, which means a cartoon. So there's a cartoon that is created after me, or is based, like the character is based on my life as an ocean scientist, and her name is Nitro. This is actually the old cod. Nitro has evolved. They now have a new version, which looks more real. And I'm also part of Ocean Women, which is a fellowship at the University of Cape Town that uplifts and funds Black women and women of color in ocean science. So that has opened up so many opportunities, has also put us, when I say us, I mean myself and the cohort. One of them is in here now, Faith. And it has introduced us to so many role model women in different industries of STEM. So now to get into the actual science that I do. So the science that I do is very much motivated by what is currently happening, which is global warming, which leads to global climate change. And the idea is that with us understanding the ocean, how the ocean was in the past, us studying the ocean now, we can predict what is going to happen in the future. And this is extremely important because the ocean takes up the most carbon from the atmosphere when compared to all resources in the world combined, even forests, all the forests in the world combined. And the ocean doesn't get much credit for this. And actually, the main thing that does this is not the actual water itself, but the phytoplankton in the water. And I love that Russell has started the chat around phytoplankton, because a lot of people don't even know what the heck a phytoplankton. People will come to me, the heck, a phyto what? And so it's nice to have had you guys be introduced to what phytoplankton are, what they do, because my science is a bit different from what Russell's science is all about, but it's all under phytoplankton. So my science, as I said in the beginning, tries to understand geochemical cycling in the ocean, especially regions that are currently undersampled. And when I talk about regions that are currently undersampled, we're talking about regions like the Indian Ocean, the Southern Ocean, which is now having a number of researchers flooding towards it to try to understand it. But in terms of research, only 5% of the ocean has already been studied. So when we talk about the Southern Ocean, it's just a speck in, it's a drop in the ocean. It's just a drop in the ocean. The amount of research that has already taken place. So my work tries to understand these regions and tries to understand the microbial interactions with the nutrients, because without the nutrients, the phytoplankton they cease to exist, because phytoplankton needs sunlight. They also need nutrients to survive. So my science looks at these different phytoplankton, particularly Synechococcus, which is a type of small phytoplankton and small eukaryotes. And some of these eukaryotes you can refer to as diatoms. But the tiny ones are the ones I'm interested in. So the science that I do basically looks at nitrogen, as I already said, and we are interested in two main nitrogen isotopes. And the basic idea is that nitrogen has two stable isotopes, N15 and N14. N15 is heavier than N14, and N14 being the far more abundant isotope in the environment. So the ratio of N15 to N14 in a sample is quantified as a delta N15 in per mil notation over here. This is how we quantify these in a sample. The higher the N15 relative to the N14 in the sample, the higher the delta N15. So this is quite important in the science that we do. And the different nitrogen sources can have different delta N15 notations. So when I talk about the different nitrogen sources, we are talking about ammonium and nitrate, because those are the main nitrogen sources that my science looks at. Because when we see that, okay, this sample has a lot of nitrate, we equate that to the region taking up more carbon than when it has more ammonium. So this is why my work is so important and so fascinating, because we, from our samples, can tell what the region is doing just by looking at these particulate organic nitrogen isotopes. So as I said earlier, my regions of interest are the ones that are currently undersampled. And my particular interest is the ASCA array, which is a transect between, I'm actually in Port Elizabeth right now. I'm not in Cape Town. So I took some time away, and I'm at my sampling site right now. So this ASCA array is between a region here in South Africa, which is between Port Elizabeth and East London. And this is in the Indian Ocean. So I'm interested in understanding what happens along the coast, because when we sample, we check what is happening along the coast, we check what is happening within the current itself. This is the Agulhas current. And then we also look at the open ocean. So I want to see how these regions differ, because they are environmentally different. And to carry out this work, my work is very lab-based. It's very chemistry heavy. And to carry out the work, we first have to sort these phytoplankton communities. So we sort the Synechococcus and the small eukaryotes into different communities, and we use a machine known as a fax machine. So it's fluorescence activated cell sorting, which is a flow cytometer. So we put the sample into the machine. The machine sorts the communities according to what we tell it to do. We call it gating. So we tell it you gate these according to how they fluoresce. If they fluoresce between these wavelengths, put them this side. If they are fluorescing between these wavelengths, put them this side. And that's how we separate them. And anything that does not fall between those goes into the waste. And then this allows us to then take these samples and do a technique known as the Denitrify method, which is a scientific, a very chemistry-based scientific method that allows us to see how these different types of communities have taken up the different nitrogen sources. And as I said before, the ammonium and the nitrate, you will see from the different communities whether this community has taken nitrogen or some of these community members have taken ammonium, it will all be vivid and will be telling a story of what is happening within those waters. So here you are looking at plots of these small eukaryotes after sorting. So what the story here is telling is that the small eukaryotes in green, if they are close to the purple line, which is the nitrate, it means that this part of the region and this, you remember I talked about the coast, the core of the current, and the open ocean. So you see that in the core of the current, the green circles are very close to this purple line, which means also close to the coast, the region is taking up a lot of carbon, which means it's storing the carbon, it's pumping it into the deep ocean. This is a good thing. And then in the core of the current, you see that, yes, even here, the same thing is happening, while in the open ocean, less of this is happening, even though we see that in the surface waters, we can observe that the eukaryotes are taking up the nitrogen. And so that means even in the open ocean, there is some form of drawdown that is happening of carbon. And this is very important because this means it's not just happening in the transect that we have sampled from, it's probably happening across the entire Agulhas region. And the Agulhas region is massive. And the Agulhas region plays a huge role in the weather and climate of not just South Africa and Africa, but globally, because some of its waters go into the North Atlantic, and that means it has a global impact on the global ocean. I also calculate the nitrogen and carbon uptake of the ocean, where I go to the ocean, I sample, I come back to the laboratory, I conduct the lab tests, and then the data that I get, I have to analyze. Of course, there's a lot of coding in R, frustrations, my goodness. And then analyze the data. And the data basically will tell me or tell us what is happening within this region, because they give us specific information about how much the region is taking up, how much carbon, how much nitrogen is being taken up by the region per hour, per day. And this is really, really cool because it paints a picture, it gives us perspective when we combine these data with the data that we have in our other study of what types of phytoplankton are doing the work, and now understanding at what rates. So I love being a woman in ocean science. I actually take advantage of it in that I try to also, what they say about women, when you educate a woman, you educate a nation. So that's who I am. I go around talking about my work. I do TV shows. I write. I've been asked to write articles for The Conversation Africa. And that's a big platform. The editor sent me a message telling me this is the first time we are seeing a science article making so much, basically getting read so much. I don't know how the writers say it, but that was a lovely thing to hear. It means that the work that we are doing, when I say we, I'm talking about myself and my supervisor, Dr. Sarah Fawcett. The work that we are doing is reaching this, and it's educating, it's changing minds, it's letting everybody know that you can do it. Kolisa is doing it. She comes from this background. And I mean, science is fun. It's something that anybody can go ahead and do. But also there are difficulties being a woman in science. And some of them I've listed here, but one of the difficulties I really have a problem with, because I'm a Black woman, I have a huge ass. So when we go on these field trips, you have that just don't fit you, they just don't fit. They are not made for you. But women are curvaceous, and it doesn't matter whether you're a Black woman or it doesn't matter. We need to have clothes that are made for us, because we are now in these spaces, we are in the field, we have arrived, and everything has to suit us, has to accommodate us. So that's me.
We've had a few questions come into the Q&A box while you've both been talking. So perhaps I can start by directing the first question to Russell. You were talking about the harmful algal blooms, and our question is there a way to reverse the effect of these algal blooms?
There's not really a way to reverse the effects. There's no kind of, well, the toxin, from what I know, there's no kind of like antidote to that toxin. What you can do, and this is the research that I'm looking at, is the same way as if you were going to go out and measure any kind of plankton, you would measure the temperature and the salinity and the depth, that you found it at. There's now this school of thought that how turbulent or how mixed the water is plays just a key, as much as an important role as the temperature or the salinity in determining what phytoplankton species can live there. So I was saying, a lot of toxic plankton have a little gas bubble inside them called a vesicle, and they're positively buoyant. So that helps them get up to the top where the sunlight is, and that helps them to bloom. So by mixing the water column up, they lose their competitive edge and they're replaced with a non-toxic species. So a lot of the work that we're doing at the University of Bath is trying to see the most efficient way to mix reservoirs. And there's things like, as well as the pipes, there's also like these big ceiling fans that like push the water down in these big convective cycles. There's subtle ways that you can change, whether the bloom happens or not. But a lot of it is down to around coastal areas, as around to eutrophication and just farmers putting too much fertilizer, and that being all like sewage overflows and things like that, which puts extra nutrients into the water.
So I was reading something the other day relating to reservoirs where they use black plastic shade balls to stop the light getting down to the plankton. Obviously, that's not something that you could do in the ocean, but is that a workable solution for lakes or reservoirs?
Yeah, I think obviously there's this balance, isn't there? And I'm not sure. I mean, a lot of the time with ocean chemistry, the discussion is around plastic, and I'm not sure what kind of like interesting chemicals might be introduced into a drinking water reservoir through these black plastic balls being photochemically degraded under UV light out in the sun. It seems to me to be like like the old woman who swallowed the spider to catch the fly. If we're just like, oh, okay, well, why don't we just like stop have a bit more control about what we're putting on our fields, instead of having to then invent another thing to deal with the symptom. It's like a cause and effect. So
maybe let's deal with the cause rather than try to mask the symptom.
But the black balls thing does look cool. Like, it's like, yeah, this is cool, isn't it? Look, a whole lake full of floating black balls. Like, there's a great YouTube video about it, which is, I saw it. And obviously that all went around my department. Everyone was like, yeah, I'm not so sure about this. But yeah, it's I guess like an interesting thought experiment. And, let's see what happens.
We've got a couple of other questions. One for Kolisa. So is the trend showing an increase or reduction of the absorption of carbon, according to your research?
So the trends are showing that the agallis current is actually doing better than we thought, because because the agallis current is oligotrophic, which means it's low in nutrients, we thought it wouldn't be taking up that much carbon. It wouldn't be drawing and pumping so much carbon to the the ocean's interior, but whoa, we were shocked, especially around the region of the Agulhas current. We see that the current itself has an impact, like a huge impact on how much carbon is being taken up, and it's a lot. So the trend is showing that there's an increase. And now we are just worried about what will happen in the future, because the waters are warming, and we know that with the waters warming, the shift will happen. I see that the follow-up question is asking about the species that are there, what types of phytoplankton that are there. And we see that in the Agulhas current, like I showed in my slides, the small eukaryotes, which are the bigger according to that region, because the region has very little nutrients, the big guys are still small. So the big guys, which are the small eukaryotes, are the ones that are carrying out the uptake of the carbon. And this is a trend we see even in the North Atlantic. It's not exclusive to the Agulhas current. However, in the Agulhas current, we see that the uptake is much higher than the other regions. The Agulhas current is fascinating. I want to work on it forever.
It sounds absolutely fascinating. I'm sure there's so much more to learn. But we do have one more question. I know we've got quite a few science students in the audience today. And somebody has asked, do they need to be good at biology, chemistry, and physics to be able to get into marine science? Or are there different ways of studying the oceans without being good at all three subjects?
In oceanography, so when you talk about marine science, you talk about a vast majority of subjects in marine science, right? So I'm doing oceanography. Somebody else might be talking about marine biology, for example. In oceanography, you need physics, because we do a lot of physics. Ocean physics, chemistry for my field, but oceanography is vast as well, because you might be doing ocean modeling, but everyone has to do the coding. And so mathematics is very key. And of course, I mean, if you have a bit of biology that you understand, that helps. But when we come to, say, for example, marine biology, biology is most important, as compared to physics. But then when we talk about marine science, we're talking about a range of subjects, of modules within marine science. So I would suggest that somebody would have math, physics, chemistry, biology, all four.
So Russell, what would you like to add to this area of discussion?
Well, I would say that I didn't do any science A-levels. So I actually started doing fine art painting. And so that was my background before I became a marine scientist. I had maths and geography as well as art. So when I decided that art wasn't for me, I was able to switch to oceanography.
Somebody else has asked, what about geology and the slow part of the carbon cycle?
Well, I guess there's this idea that plankton happens, and then as it sinks down and it dies and it sinks into deep ocean, it gets turned into sediments quite over time. I guess in terms of looking at whether we can alleviate climate change using this process was a thing that was originally floated around in about 20 years ago, where they were going to go out into the oceans and fertilize them with iron, which is a micronutrient. Yeah. So a lot of the time they saw that all the world's oceans had all of the nutrients. It was like, why isn't anything growing here? And just if we could go and sprinkle iron all over the world's oceans, then it would cause these plankton blooms, which would then draw down even more carbon dioxide and trap it into the deep ocean.
He's quite correct. As we've seen, plankton blooms, they could have devastating effects on the ocean. Additionally, when we sprinkle iron in our waters, we basically alter the natural state of the waters. So the regions that would need that needed, for example, the Southern Ocean, which is close to Antarctica, and Antarctica is covered by ice, and the ocean needs iron, and iron comes from dust. So if there's no dust coming from the continent, these scientists that Russell was talking about thought, okay, what about if we sprinkle some iron, and that was a terrible idea. We alter the natural state of the waters, and it's just not good for the phytoplankton. But also in terms of the question, the original question, the geology part, in my work, we do a lot of geology plotting. So the sketches, I could say visual data representation of mine has a lot of geology in it, because you have to show the where the land is under the water within your code. So it's one of those things I struggle with actually with my code in R, where I have to enter the code for the bathymetry, because it has to show where it starts. We have to see where in terms of the carbon cycle, where the land starts, because that shows us where most of the nutrients come from, because close to the land, we have more iron, more nutrients, and therefore the carbon cycle, which links to the question that has just been asked, is stronger.
Great, thank you, Kolisa. There's just one last question. Somebody has asked, what is the biggest predator of phytoplankton, or what eats the most phytoplankton?
What eats the most phytoplankton is probably, I think, anchovies, because they just swim along with with their mouth open and let all the phytoplankton swim in. Larger things like whales and whale sharks and basking sharks, they tend to filter the larger stuff like the zooplankton, which eat the phytoplankton. So yeah, I'd say probably you're looking at those kind of like small fish which would filter feed those.
But there are a lot of them, aren't they? They travel around in huge shoals.
Yeah.
Well, we have come to the end of our time. Thank you both for sharing your research, your insights into ocean chemistry. But thank you all for coming, and good night.